at Children's Hospital in Cincinnati to build the concept of circadian medicine as something that's distinct from, but also encompassing sleep medicine. So first, I'm going to give you an introduction into the core oscillator. Then I'm going to tell you something you already know, which is that hospital medicine is itself rhythmic. Everything is done in a rhythmic fashion. I'm going to tell you about a dosing time of certain medications mattering, and how we're building circadian medicine in our buildings, exploring those rhythms with the electronic medical record, and how we're learning about the clock from patients with rare and circadian disorders. So our field got its start in the late 1960s, early 1970s due to the pioneering efforts of this geneticist Seymour Benzer. So Dr. Benzer and a graduate student, Ron Kanopka, did a mutagenesis screen of the X chromosome, looking for flies that got up a little earlier every day, so-called period short flies. So at this point, I like to poll the audience. Who here is a morning person? They like to get up early. Personally, I find you guys annoying. But they found early fly types. They also found late fly types called per-long flies. So who here likes to stay up late? I want to party with you, cowboys. And even flies that were completely arrhythmic with no 24-hour rhythms at all. And when they did the genetics at the time, they realized that all three traits mapped to the same locus. And in a series of papers in circa 1994, the Hall, Rosbash, and Young labs cloned the gene. And it was the period locus and the first member of the PAS domain containing transcription factor family. Other PAS domain families include the hypoxia-inducible factors, which is probably familiar to many of you in the room. But it all started in the fly. All right, so who here has learned about rhythms because they had a child? Raise your hand. Yeah, we mostly forget that we're rhythmic until we do something like have a kid. So this is actually the actogram activity of an infant from age 3 weeks all the way through 26 weeks. And you can see that at the beginning of this child's life, they're almost completely arrhythmic. And then around week 7 or so, they start to develop an ultradian rhythm, a rhythm longer than 24 hours. And right around week 20 or so, week 16 maybe, or 17, this particular infant snaps into a circadian rhythm where they're waking up about 8 o'clock in the morning and they're going to bed about 8 o'clock at night. So I actually turned this into a greeting card. Me and Maya Buchin, a colleague at Penn, and when somebody had a baby, we would write, "You are here," right around week 3 or 4. And then this is when life gets livable around week 16 or 17, just to remind them that it's a living hell, but it's a temporary living hell. Now, one of the good things is because you're sleep deprived, you don't really form memories very efficiently. So you forget how horrible it was. And in a few years, you have another child. At least that's what happened to me. It's not just human infants that have rhythms. It turns out most creatures on Earth have a circadian oscillator. Even cyanobacteria, fungi have circadian rhythms, even though they have a doubling time much shorter than 24 hours, because it confers an adaptive advantage to all these organisms. Charles Darwin here, this mouse. The clock is an adaptive advantage to organisms. You don't have to be on all the time. You can sleep or rest or do whatever some of these organisms do. But this is why a lot of students, trainees care. Raise your hand if you're much younger than about 30. I'm not. Put my hand down. So this is actually looking at two aspects of sleep. This is not just the timing of sleep, but also the amount of sleep that your body requires. And so this is looking at kids all the way from age, say, 16 or so to adults all the way to 80 or so. And what you can see is that on people's free days, typically weekends, the younger people tend to rest a lot more. They're sleeping about almost 8.5 or 9 hours on the weekends. And during the week, typically during school or work, they're sleep deprived about an hour and a half or 2 hours a day. Now I'm 55, so I'm actually sleep deprived only about 30 or so minutes per day, including last night. There was a lot of ambulances. You people are so rude with all your taking care of your patients and all that jazz. They were just coming left and right last night. But really it's the interaction of these two systems, the homeostat, which really tells your body how much sleep it needs, and the circadian system, which makes sure that you get it at the right time. Now, it's not just sleep, it's also all these other aspects of your physiology, things like breathing rate, body temperature, skeletal muscle function, lots and lots of different hormones are all under circadian control. I don't think I stuck the slides in today, but we just measured 200 lab values, and something like a third of them are influenced by when you actually sample the actual sample. So I'll illustrate it with blood pressure. This is ambulatory blood pressure monitoring. Who here has done ABPM on themselves? So like maybe 5 or 6 people in the whole room. So this is really the gold standard for a hypertension. 10 or so years ago, I went in, my then at the time twins were 4 years old. And so I got measured every 15 minutes during the day and every 30 minutes at night for 2 consecutive days. There's a couple interesting things here. The first is that was the twins' bedtime, and I wasn't even putting them down. I was just listening to them caterwauling about it. And so that made my blood pressure go up. And the second was, even though it was a very, very civilized commute at Penn, just the thought about going into work would send my blood pressure rising pretty much every single day. Yeah, the Ivy Leagues. Who knew? Yeah. All this biology is driven by these core molecules. Jeff kindly pointed out that I discovered BMAL1, which is a core transcription factor of the clock. BMAL1's partner is clock, which was discovered by Joe Takahashi. And then there's a second gene called NPOS2, which can also pair with BMAL. Those two are transactivators, BHLH pass domain containing transactivators. They bind response elements throughout the genome, hundreds of different response elements, but notably in the promoters of the period and cryptochrome genes. The PER and CRY proteins are translated in the cytoplasm, where they complex with each other. They're actively imported into the nucleus, where they reach a level where they can physically interact with clock and BMAL, pull clock and BMAL off of DNA, repressing transcription again at hundreds of loci, but notably their own loci. And then later in the cycle, the repressors are actively degraded by FBOX binding proteins. And when the repressors are degraded, that lets clock and BMAL hop back on DNA and initiate a second round of transcription. So we think about this as a transcriptional translational feedback loop that's pretty much operational in every single cell in your body. It may run weaker in certain cell types like immune cells. We were just talking about that. Curiously, it's actually not super strong in brain cells either, neurons or astrocytes, but it's pretty darn strong in things like the liver, the lung. Many of your peripheral organ systems have very strong autonomous clocks. That means you can take the lung out of a mouse, put it in a dish, and it'll spontaneously oscillate. Now, clocks are accurate, but they're biological systems. They're not 100% accurate. So they need to be reset. The principal resetting cue in mammals is the light-dark cycle. And so light at certain times of the day, early in your activity phase and late in your activity phase, is when the clock system is most sensitive to light. There's 2 classes of photoreceptors, the image-forming photoreceptors that you're using now, and a second photoreceptor called melanopsin, which is a blue light photoreceptor. It actually is most sensitive at 480 nanometers. And then both these systems signal to the SCN neurons and let them know that the lights are on or the lights are off. And that helps keep clocks running pretty accurately. So at this point, I always ask the audience to take out your phones. You know you want to. If you go under settings, display and brightness, there's a mode called night shift mode. I have mine turned on 10 p.m. to 7 a.m. And what that does is it takes a little bit of the blue out of your screen. It might get you a few extra minutes of sleep. This is probably the most useful thing you're going to learn from my talk today. So if you want to pack it in, I won't be upset. All right. So a few years ago now, we were interested in looking at rhythms in the hospital record. And so our first protocol that was approved was to look in the intensive care unit. And so we went to our colleagues, Ted Cooper, Amy Shaw, and Dave, my partner in crime. And we began pulling records from the EMR, from Epic. And so we looked at a number of different drugs, about maybe the top 10 most commonly used one in the ICU. This is an example. This is using hydralazine, which is used in acute care to lower blood pressure. It's available orally, but also you can give it intravenously. And so this is looking at the number of orders placed for hydralazine on the top, and then when the actual hydralazine was given to the patient. So the order takes a little bit of time, it goes down to the pharmacy sends it back up, then it's about an hour or two delay between ordering the drug and getting them on board. And what you can see is that there's a huge spike in orders between say 8 o'clock in the morning and 10 o'clock in the morning. And you all know why that is. That's your rounding period. It's probably the case at every single hospital in the Western world that they're prescribed with this sort of regular pattern. And there's a second rounds in the afternoon between 4 and 6 p.m. So there's another burst of orders 4 and 6 p.m. And then the orders go to the pharmacy, they're filled, and they're finally given to the patient. And so you can see there's a spike in when the actual drugs are given. Now at this point, we had a little debate in the lab about whether or not this was going to be true for all drugs. I argued that it was going to be true for everything. And some people thought it was only the blood pressure medication, because there's a long literature on control of blood pressure and the circadian system. But when we looked at the 12 most commonly used drugs in this inpatient setting, they all basically followed the same pattern of a big spike in orders around the rounding periods. And then the first doses of these medications are given with about an hour to 2 hour delay. And for most of the medications, it probably doesn't matter when you give them. So this is probably fine. But I picked out a couple of examples where it probably does matter. The first is pain medications, morphine here and acetaminophen. But patients are selectively in pain in the morning. In fact, they complain most about pain at night. And so here's an opportunity for our system to get better by asking about pain in night and making sure that they're not untreated for pain for long periods of time. The second example is in the upper right-hand corner, and that's the use of vancomycin, which is a last-line antibiotic used to treat sepsis. And so it's also given with this morning spike and afternoon spike, but you should probably be giving vancomycin at the first sign of infection, not the next day, because that actually can kill you. And so here's room for improvement. And I'll leave you with a final thought, which is we can also look at efficacy using the inpatient clinical record, because we do measure things like blood pressure pretty often, every couple hours, if you're in the ICU setting. There's a huge opportunity for doing research on this type of data, and almost nobody's actually looking at it. So it's not just there for billing. I know that's important, but it's also a research tool. So our next steps are really to analyze more drugs, start looking multi-site. So we've done some work with Jay Huey at Vanderbilt, Phyllis C at Northwestern, exploring some of these rhythms in drug orders and drug administration. And of course, it's the same everywhere. And then looking at different types of readouts, looking at the effect of age and sex and other comorbid conditions on how these drugs are both prescribed and used and how they work. So the next vignette I'm going to tell you is about it actually mattering. And so this is work with my postdoc, Mark Rubin, Dave Smith, my partner in crime. He's a sleep board at ENT, and my old division chief at Penn, Garrett Fitzgerald, who's a world-renowned cardiologist. He developed low-dose aspirin therapy. And I'm actually on low-dose aspirin therapy right now. I'm in that little group of people around age 55 where there's benefit. My blood pressure is doing great. Thanks for asking. Yeah, I really appreciate your concern. So we started to look at the last 70 years of research. So it turns out people have been looking at the effect of timing on how medicines work for more than 70 years. And so we collated all of the studies. I think we're up to 140 something studies that we've analyzed now. And these are very commonly used drugs for many different things, but there's a whole bunch of studies on hypertension, cancer, asthma, dyslipidemia, arthritis, et cetera, some of the most common maladies in humanity. And when we looked at the governing principles of whether or not it had an impact on how the drugs worked, it turned out that drug half-life was really the dominant force. So if you have a half-life much less than, say, 8 hours, it's extremely likely that when you take the drug is going to impact how well that it works. And conversely, if you have a half-life of over, say, 30 or 40 hours, it's hard to imagine how when you take the drug actually has any impact. And of course, we don't see that it does have any impacts. So it looks a lot like the short-acting drugs, which is probably a quarter of all drugs, are going to matter as to when you take them. And yet there's been very little research on many of these classes of medications. 100 trials in 70 years is not very many. So we've also spent a lot of time and effort. This is work with Michael, looking at how the genome is regulated by the clock. So about maybe 15 or so years ago, Michael and I got interested in trying to formally define the best way to do these studies. And so we oversampled. We took 3 to 5 mice every hour for 2 full days. And then we pulled data out until we started to lose the power to detect circadian rhythms. And when we did that, we discovered that about half of the genome is rhythmic in at least one organ in the body. So we checked out 14 different mouse organs and about the same number of human organs. I won't really get into the human work. We can't exactly kill 3 to 5 people every 2 hours for 2 days because that's illegal and wrong. But we figured out a way to look at the phase of a sample from looking at the actual data. And many of these studies, especially in humans, are not just 20 or 30 people, but they're thousands of people we can spot these rhythms in. So when we did this work, we discovered that about a quarter of all drugs are short half-life and probably matter when you take them. These are some of the most commonly used drugs in medicine, period. And you can look at this list, and very few of the drugs that are on this list have a time of day dependent indication, despite the fact that it probably does matter when you take them. There's some examples here like Lunesta. Of course, you take a sleep drug before you want to go to sleep. So that's pretty obvious. But for the most part, the rest of them don't have a time of day dependent indication. So I mentioned that we were able to do these studies in humans. So I had some timestamp data from Colleen McClung's lab at the University of Pittsburgh, and we applied our machine learning methods to figure out what time, and that's in blue on the right-hand side. So time on the wall is in red, and time on biological time is in blue. And you can see that, if anything, our inferred biological time is a little cleaner than the time on the wall, and that's because all these people passed in the hospital. And hospitals are really terrible environments for being entrained. You've got people coming in the room all the time. The lighting is often very poor. The television is terrible. I spent about 6 days in last year, and I just had an absolutely horrible time. You're all terrible people when it comes right down to it. And the food, oh, geez, not good. Not good. I know. Is the food good here? I see one head nodding. So the long and short of it is the way how we can do this is taking advantage of a principle, which is if you have 2 out of phase waves. In this case, the 2 out of phase genes are ELOVE L3 and UBXN1, and you plot them X versus Y, they form a circle without regard to when you actually take those samples. So you have to take my word for it, but take 2 waves, plot them half pi out of phase, they're going to form a circle. And so we were able to use this principle and reconstruct output, 24-hour genetic output. In this case, this is in human liver. I think it's like 300 or 400 human liver biopsies here. And here are some drug targets: AGTR1, DDC, the target of the dopamine decarboxylase inhibitors, PDE4, PPAR, PPAR gamma, xanthine dehydrogenase, et cetera. So there are these really strong rhythms in all these drug targets, and yet almost none of these medications are taken with a time of day dependent indication. So I'm going to argue that we're at the time now where we can begin to think about time as another way to achieve precision medicine, not just genetics. How many of you have had your genome sequenced? Like a couple people in the room. 1, 2, 3. That's the most I've ever seen. I do this talk a lot, and that's the most I've ever seen. But it's not in wide use in medicine. Maybe they'll check out your genotype for certain P450s, but we really don't use genome sequence. So I'm going to argue that circadian time is probably more useful because you can figure out somebody's phase with a simple questionnaire. The microimmunic chronotype questionnaire is just 4 or 5 questions. When do you go to sleep on a typical night? When do you wake up on a typical morning? That's more or less, that's it. So at Children's Hospital, we're applying these principles to many aspects of our care, including circadian aware lighting systems, our operational procedures like the timing of feeding and when to dose medications, and then ensuring that our patients with unrecognized circadian disorders are seen by specialists. And I'll tell you a little bit about each of those projects. The first one is we built a new critical care building at Children's Hospital. It's $600 million, 225-bed critical care center. And about 80 of the beds, the NICU beds, have circadian aware lighting systems where we can change not just the intensity of the light all the way up to a pretty bright 10,000 lux, but also the color of the light. And I tried to argue that we should have also done this for the staff, because the staff are, if you're working the night shift, a little bit of blue light before your trip home may keep you more alert and less likely to have accidents. But we only had the money to do the NICU bed, so that's going to have to wait. We have 20 portable units, so that might be possible to do some staff research along with the patients. So light's a really important cue, but another important cue is feeding. And so I was walking in the hospital, and I ran into Stella Davies and Chris Standoid. So Stella runs one of our cancer units, the bone marrow transplant unit. And I asked, would it be okay if we just walked down to take a look at the rooms? And so she was kind enough to take us down there. You have to scrub up because these patients are pretty vulnerable. Leave your electronics behind. And I walked into the rooms, and of course, the lighting was absolutely horrible. Different tiny little windows in the hospital unit, and then tons and tons of gadgets with all kinds of lights on them, many of which were the exact wrong color, blues and greens, that are very hostile to your body's light sensitive system. But the thing that really surprised me was learning about how people are fed. So after you have the procedure to get rid of your immune system, you can't take in food orally. So you have to be fed by total parenteral nutrition. This is where all your calories, all your minerals, everything's coming in on a bag. And I learned that they fed people 24/7 for at least a minimum of 5 or 6 weeks, but even some patients as long as a year. And what we knew from the literature was that even if you make healthy undergraduates eat at night, that they develop problems with blood pressure, early signs of metabolic syndrome. So, and I asked, and it turns out some of these BMT patients also have hypertension and things like that. And so we asked a very simple question, which was, did we really need to feed them 24/7? And so we wrote a protocol up, and we worked with the nutritionist, Sydney, who came up with a formulation of the food that let us fed them over a 12-hour period instead of a 24-hour period. So just concentrated the food down. And in addition, we gave an extra 2 hours if anything were to happen, if we weren't able to get enough calories in. We didn't really need to do that except for one patient whose line came out. And so we had to put the line back in, and they got an extra 2-hour extension of their feeding time. But everybody else has tolerated the 12-hour feeding period. And I'll cut to the chase, but if the lower left-hand corner here, you can see the IV nutrition time. And we had two groups, the daytime restricted and the continuously fed groups. And it looks like the people that are in the daytime restricted group are able to orally feed about a week ahead of their peers that are fed 24 hours. So anyone want to take a stab at how much money it costs per day in the BMT ward? Take a stab. How much money do you think it is? Yeah, go ahead. A day? That's why there's so many buildings going up here. Geez, you guys are raking it in. Yeah, it's about $10,000 to $15,000 per day in the bone marrow transplant ward. And if we're able to get people home a week ahead of time, that's going to end up being about $75,000 per patient. There's 3,000 to 5,000 patients per year in the United States that need this procedure. I did the numbers, and it ends up being about $300 million you could save the US taxpayer simply by feeding people at the right time. So I think this is a huge opportunity for collaboration, because we need to go multi-site. And I think there's a huge potential here for getting kids and adults home and with their family members sooner by feeding them appropriately instead of 24/7. So our lab studies the clock and sleep at several different levels. In the upper left-hand corner, that's a U2OS osteosarcoma cell line that we've engineered with the firefly luciferase gene. And that's actually continuous data. That's measured every 10 minutes for 1, 2, 3, 4, 5 days, but we can keep them going as long as you want. Lower left-hand corner is the PER2 luciferase mouse. So this is a mouse that's been engineered with that same period gene from flies, the promoter of the period gene driving firefly luciferase. And you can impart this rhythm onto a mouse. In fact, this is the SEN that's going in the lower left-hand corner. But you can also look at rhythms in the lung ex vivo, in the liver ex vivo, heart, whatever you really are interested in. It's a super useful model. And that's from the Takahashi lab, and they're now available through the Jackson laboratories if anybody's interested. And then there's two ways for us to study rhythms and behavior in mice. One way is using running wheels. These are very similar to the ones that have it at every pet shop, except for we hook ours up to computers. It turns out those little suckers love to run. So if you just put, actually Yoga Meyer at Leiden University in the Netherlands put a running wheel in her yard and just watched it with a camera. And it turned out that all kinds of animals walked into the yard, hopped on the wheel and ran for a while. And then they hopped off and went home, including like frogs and mice and all kinds of different creatures. They just like running. I personally don't like the Peloton. I find it a little tedious, but those animals love it. And then there's a piezoelectric system in the lower right-hand corner here. These are also possible to measure with people. You can put these films underneath a mattress and they can actually sense breathing. And from breathing state, we can infer that somebody is sleeping or not sleeping. And some people argue that you can even get at the stage of sleep from measuring the breathing pattern. I'm a little more dubious of that, but if they can get it to work, great. I'm going to argue that sleep timing is poorly understood. Most of what we've learned about sleep timing is due to the pioneering efforts of Yinghui Fu and Louis Potacek at UCSF. And so over the last 20 years, they've been studying families that were initially at Utah. In fact, they were just the subject of a Time magazine piece where there's a large family in Utah where they only need about 5 hours of sleep a night, and you can actually sleep deprive them over a period of days, and they don't seem to have any performance deficits. We found a second family in North Carolina with Allen Packett Penn where you can actually sleep deprive them, and they don't seem to have any performance deficits either. And so these were both mutations in DEC2, so these are short sleeping families. We've also found phase disorders, advanced sleep-wake phase disorder, that's the new name for it, ASPWD. I don't really like it. I like the old nomenclature, but they didn't ask me. And there's 4 genes that have been found that lead to advanced sleep phase, and 1 gene that's been found that leads to a delayed sleep phase. That was found by Michael Young and his colleagues at the Rockefeller. Some of these issues have fallen into popular culture. So this is where I'm going to give you a little Southern rock lesson. So in the early 1960s, the Beatles came over. They had a huge impact, and a whole bunch of people decided they wanted to learn to play guitar because they wanted to have the same accolades that the Beatles did. And at my high school was Don Felder of the Eagles, who's the lead guitarist of the Eagles. And Don lived in the same neighborhood as Stephen Stills and Tom Petty. And he had gotten a guitar at the music shop on University Avenue. And he decided that he was going to teach both Stephen Stills and Tom Petty how to play guitar. And so all 3 of those guys went on to form incredible bands, like Tom Petty and the Heartbreakers, like the Eagles, et cetera, et cetera. Just up the road was another band called Leonard Skinner, Jacksonville Lee High School. And they covered a song by Merle Haggard called Honky Tonk Nighttime Man. And so this is really how it goes. I'm a honky tonk nighttime man. I can't stand no light. I get my rest in the daytime and I do my running around at night. So here's somebody that's telling you they are a night person. They do not like to be awake during the day, and they like to be active at night. I had the blues this morning. I had the blues all day today, and they wish a tornado would blow those blues away. That's a terrible idea. Growing up in North Florida, I've seen what a hurricane and what a tornado can do. So that's very unadvisable. And then when my heart starts beating, when the sun starts sinking low. So they're recognizing not just that they're tired and they want to sleep in the daytime, but they're also sensing this physiological change, which is their heart rate and blood pressure is anticipating when they want to be awake. I'm going to argue that there's a huge opportunity to look at the genetic basis of sleep in kids. And that's because when you're 4 years old and it's 2 o'clock in the morning and you don't want to go to sleep, your parents are going to probably tell their general practitioner, and eventually you're going to end up in a sleep clinic. And whereas if you're an adult, and if let's say you're Jeff and you want to stay up till 2 o'clock in the morning, no one's going to tell you what to do. You can do whatever you want. You're an adult. But when you're a kid and you're compelled to do that, someone's going to be contacting a specialist sooner or later. There are also many genetic disorders. This is by no means a comprehensive list, which range from ultra rare disorders like Smith-Kingsmore to metabolic disorders like Sanfilippo disorder, Smith-Magenis, which is these patients actually have a flipped activity cycle. Even their melatonin is flipped, and it's possible to actually get them to be diurnal, and you basically have to use a beta agonist and an exogenous melatonin to flip their activity cycle, but it's doable. And then there's less rare disorders like Angelman's and Rett and genetic epilepsy, Downs, where a ton of those kids have sleep issues, many secondary to obstructive sleep apnea. So they require somebody like a Dave to go in and take the tonsils and adenoids out. And then autism, for whatever reason, autism, and there's many kids with autism that report sleeping problems. And then the big one, which is delayed sleep-wake phase disorder, which is something like 1 in 10 of all adolescents will have problems with sleep timing at some point during their teenage years. So here's an example of a 14-year-old that was in our system, and she was going to sleep at 6 a.m. and she slept until about 4 p.m. every day, which made attending school very difficult. And so with a combination of light therapy, so basically getting light early in the day and later in the evening, cognitive behavioral therapy, which is as effective as many of the somnogens that we use in the clinic, a phase shifting dose of melatonin, and then clonidine, which is a hypnotic. It's actually a blood pressure drug, but where one of the side effects is it makes people sleepy. And this is a big one in pediatric medicine because it's a very safe drug and you can use it at a very, very low concentration. So this is a mainstay for children's sleep medicine. We were able to toggle her back over to 11 p.m. to 7 a.m. sleep. She went off of the clonidine, flipped right back over to nocturnal again. We put her back on the clonidine. We've actually done that twice, and she's currently going back on the clonidine, and just so she can complete school. Once you're past your school years, it doesn't really matter. You could use this as a huge advantage. Being awake at night could be a big advantage. You could come work at the WashU emergency medicine department. You could trade the Tokyo desk in New York. There's lots of careers for people. You could be the next Merle Haggard or Leonard Skinner. All those are options for you, but she probably needs to finish school before she can really start to go down those paths. I'm not going to go through. We have candidates. We are making the mouse. Here's an example of one of the genetic disorders I told you about, Smith-Kingsmore syndrome. It's a rare form of autism caused by activating mutations in the mTOR gene. So mTOR is the mammalian target of rapamycin. There's tons and tons of problems in these children, including intellectual disability and developmental delay, megacephaly, and seizures. Of the 150 patients, the 3 or 4 that we've tracked that have passed have all died from seizure disorders. And we and others are using rapamycin and other rapalogs, mTOR inhibitors to treat this. It's been extremely effective in actually lowering the stimming behavior. These are stereotypy movements. They'll slap themselves, they'll hurt themselves, and it's been really effective at treating that. But initially, when my partner, Carlos Prada, was treating this particular kid, the parents noticed that there was a severe sleep problem. And so the dad wrote, we observed a sleeping pattern of 7 to 10 days of 12-hour sleep a night, which is, I guess that's good. 1 to 4 days of 7.5, which is, I guess, pretty decent. And then a week or so of really terrible sleep, about 2.5 hours a night. And then another okay week of 7.5 hours. And the dad noted that each cycle lasts around 24 days. So to us, that sounded a lot like delayed sleep-wake phase disorder and a patient toggling over to the next day. This is where things get weird. The patient was in Cincinnati, in Liberty, Ohio, which is about 9 miles north of our hospital. But for whatever reason, they wrote a sleep doctor at the University of Montreal who sent the note to Michael Rosbach at Brandeis University. Michael Rosbach works on Drosophila. Well, don't know why they sent it to Michael Rosbach, but they did. He sent it to me. I handed the contact information to Dave, who's in the office next door. Dave realized this was one of Carlos's patients. Carlos was about 50 meters up a couple floors and a little bit over. And we realized we had never actually talked about sleep. So we had been working with a collaborator, Andrew Liu, on the mTOR system. We showed first in vitro assays that we could add rapamycin and make the clock go long period. We could do this genetically by knocking down mTOR with short hairpin RNAs. We could use our SCN knock-in mice to monitor rhythms, and they were also long period. We looked at locomotor activity rhythms, and those for the mTOR heterozygous knockout mice, and they were also long period, both in constant darkness and in constant light. And we were able to show that you can use the telemetry system to look at locomotor activity, and the mTOR loss of function mice have reduced locomotor activity in the active phase. And we even put in the allele for one of our patients into the mouse and showed that these mice recapitulate the hyperactivity phenotype, the stimming type of behavior that the patients have. So our treatment model is now is now what was probably happening is we were using too high a dose of rapamycin. We were using the organ transplant dose of rapamycin, and that's probably too high. I asked a number of the transplant surgery team, do you often see problems with patients in sleep on a high dose rapamycin? And they said, yes, we do. So I'm hopeful this is going to have some implications well beyond Smith Kingsmore, but also how we're dosing many patients. And I'm going to finish with two of the most important slides. I think it's long past time that we work together. There's a tendency of many people in the laboratory research side, to stick in your office and not talk to your colleagues, your clinical colleagues, your other research colleagues. We all get tunnel vision about what we're working on. And it's long past time for us to start to work together on things like how do we feed patients in the hospital? How do we light up hospital rooms? How do we make the rooms less noisy? So there's simple things that we can do that'll make it a nicer place to be. We also have to start to get to the regulatory structure. So lobbying the FDA. Here's a bunch of clinical studies that show when you take this drug matters. How can we get this implemented on the medical side? I think one quick way to do it is using your after visit summaries. So there's an Epic plugin that lets you recommend what time to give medications. That's built into Epic. In fact, when I was in the hospital, they gave me my after visit summary, and it had a little checkbox by when to take every single medication. And I looked them up, and they were almost all right. The only one they got wrong was low dose aspirin therapy, which should be taken prior to going to sleep. But every other medication they got right in the after visit summary, and it's at your disposal. And the most important slide, this is the team. Rochelle is a sleep-boarded neurologist. She did her training at Boston Children's, and she ran the Rett clinic there. Dave is a sleep-boarded otolaryngologist and surgeon. He's in surgery on Mondays, and he's in the circadian clinic on Fridays a couple times a month. And during the rest of the time, he operates a lab looking at the intersection between hypoxia and the circadian system. He's also a brilliant speaker. So maybe several years down the road, you might want to bring him in for one of these lectures. YY is a talented graduate student. She just sadly went off to Merck. I'm not sad that she went to Merck. I'm just sad that she's not around anymore. I really like her. And then Jiffin and Danilo. Jiffin's my lab manager. Danilo was a visiting scientist from Brazil. I have a great collaborator, Andrew Liu. We've been working together now for almost 20 years. I've done some work with Eric Bittman. We just published a paper on a loss of function model of Cry1 in Syrian hamsters. Who knew? And then I have collaborators in Cincinnati from Procter & Gamble, and then Ron Anafi, who's a sleep-boarded internist at Penn and a former trainee, and Jay Huey at Vanderbilt. And with that, I'll take any questions you might have. And they told me to leave these up here on the screen. I don't know if this is like getting you a free lunch or something, but have at it.
Thank you so much for that great talk. We have time for a few questions. Tom?
So I thought a really great talk, but you had restricted eating during the day.
Yeah.
Yet the liver is one of the most entrained organs. Why wouldn't you restrict eating at night so it'd be more?
What I mean by time restricted is that's when we feed them. So we only fed them during the day.
Okay.
Yeah. It's popped into the popular culture. There's a couple other intermittent fasting is another way that people talk about it, and you've probably all seen something about that in the popular media. But they're all basically limiting the feeding window. We were limiting it to 12 hours. We don't think there's anything magical about 12 hours. It could be 10 hours is better. Could be 14 hours is better. We don't really know. But I know that if you feed somebody, even a healthy person, 24/7, they're going to get unhealthy pretty quick. And that's what we were doing. Still standard of care.
Mike?
Yes.
Mike?
I'm curious, so most of the drugs need to be timed in some way. What are the, sort of endpoints or markers that one use to guide you on to figure that out?
Well, it sort of depends on the class of medications. Like where the research has been the best has been like in areas like statins, where they're just looking at cholesterol levels. And the short acting statins, it makes a big difference in how they work. And the long acting ones, it kind of doesn't. I'm actually on one of these statins that has like a 15 or 16 hour half-life, and it probably doesn't matter when I take it. I still take it prior to going to sleep, because it turns out people only really pay attention after they wake up and before they go to sleep. So those are kind of the two times, the critical times to think about. I also take my blood pressure medications before bed. The only exception is metoprolol. I'm really sensitive to metoprolol. Metoprolol will cross the blood-brain barrier and inhibit melatonin production. So I try to take that around 5 or 6 at night, under the hopes that by the time my melatonin turns on, some of it will be gone.
So the sleep disorders you describe and the genetic associations, do those change throughout people's lifetime in terms of different phases of their age? Because, you know, we
Yes.
see people during adolescence having sort of
Right.
exacerbations,
Right.
and as they are older.
Absolutely.
So what's driving that?
Yeah, I don't think we really know. There's hand waving about it being hormonal, but we don't really have a good sense of why that is. The other aspect is sleep is very different in children than it is in an adult. And I learned about that doing a fire drill with my what at that point they were 6 years old. And one of the two twins, I was able to get him to get up and showed him the back door. This is how we get out of the house. The other twin would not wake up. He just would not wake up. I even held him upside down with my arms and carried him, and he still stayed asleep. Yeah, don't tell anybody that I did that, but I actually did. Yeah. They just don't sleep the same way that we do. They need more sleep, and they sleep deeper.
I like with our innovation center, based in Washington School of Medicine. We have a similar innovation project where we're thinking about the same intervention you referenced with interrupted night, as
Right.
well as medication distribution, noise, lighting. One thing that we're toying with is passive monitoring.
Right.
Has that been a treatment that you all have considered as we think about limiting the number of interruptions for patients during dedicated quiet hours?
For sure. It's even possible to do passive monitoring for sleep. So you can actually use these camera-based systems to look at locomotor activity, to look at movement of a bed without having anything strapped to the patient. I actually did a sleep study myself last spring, or this past spring. It should be called a not sleep study. I don't think I slept a wink. I just stood there with all this goo on top of my head and leads everywhere. Whenever I had to go to the bathroom, I had to unplug all these things and walk over to the bathroom. It was a horrible night. Anything we can do to improve that would be great. Yes, Jeff. Thank you.
With regards to patients like prevention and expectations, sometimes patients want things that aren't biologically good for themselves. So I'm thinking about Melissa Nauert's Project Nap Time intervention at Yale, where she tried to carve out in the adult ICU like a period where patients weren't bothered. And one of the things she found is that she couldn't get the patients to consent to have the televisions turned off. Because at the end of the day, they were lonely in the middle of the night in a strange place. They found it comforting to have the television on at 5:11. So, which of course then defeated the intervention.
Right.
So, like, how does sort of the patient's expectations with factor into what you're doing? You have a kid.
Yeah, I mean, I think it's probably a little bit easier because children have a tendency to at least most of the time they listen, and the parents are motivated to get their kids home and healthy. So I think that's probably an advantage of studying pediatric populations. When I was in the hospital last spring, the TV was terrible. We only got like 3 or 4 channels, and I don't even think they had CNN. It was absolutely awful TV. It was only eclipsed in its awfulness by the food.
I think Jeff just mentioned something. I wonder your opinion about it. That's naps.
Right.
So if you're sleeping on the wrong circadian phase, you think they're good or bad, or?
A short nap doesn't appear to be harmful. 30 minutes to an hour or so doesn't appear to be harmful, but nor does it satisfy your sleep homeostat. So you're probably going to still sleep later in the day as well. I like napping. There's some judgment at home about my naps, but I personally enjoy it. Yeah.
Thank you very much.